All right, thank you so much, guys, for coming. So today we are on lecture 9. Actually, we just get one more lecture for material for midterm 2. So today we'll talk about muscle physiology. I think most of the problems in the LensMat modules centered around use of energy with the muscle, okay? So we'll kind of go around that a little bit, all right? And the muscle contraction when it's occurring, what kind of is lumped in the leg phase, kind of like the things that happen before the muscle is actually contracting. So we'll kind of go through that. And today we'll talk about what happens when the muscle is actually excited, what is the physiology behind that, and then it actually contracts. All right? So that's what we'll talk about. So we'll talk about the events at the neuromuscular junction. You know, at the junction of the neuron and the muscle cell or muscle fiber. All right. So the way these action or the way muscles are stimulated or any message is sent in the body is via electrical signals called very minute electrical signals in terms of millivolts called action potentials. All right. So we'll have a few lectures that will be talking just about action potentials and electrical activity in the body. All right. And then when we do electrophysiology, we'll be able to see that in action. All right? So it is the nervous system, of course, that stimulates the muscles, okay, to contract via those action potentials. So that's the electrical conductivity in the body is via action potentials. So when the membranes are at rest, they are said to be polarized. Polarized means you have separate charges. That's what it means. It means one side of the membrane is more positive and one side is more negative. That's all. So when a cell is at rest, a muscle cell or whatever is at rest, the membrane potential is minus 70 millivolts. Okay? So that means the inside is negative. The sign on the membrane potential indicates what the charge is on the inside. Okay, so minus 70, it means inside it's more negative and outside is more positive. Okay, so that's what it means. All right. So if it's plus 30, what does that mean? Plus 30 millivolts means what's happening at the membrane potential here. Yes, and more negative on the outside. Okay, so all what an action potential is reversal of resting potential. An action potential that happens when positive ions rush into the cell, make it more positive this time, and the outside becomes more negative. So that's what an action potential is, and then it's reversed and it keeps going like that, and that's how the action potential is propagated. To propagate is to send along the nerve fiber or the muscle cell to spread the impulse throughout, so that you can excite all those proteins that contract. Okay. So that's what happens. Okay. It's just a reversal of that, and then it repolarizes again, goes back to the resting potential, and it happens again and again like that. Okay. So and we will talk a little bit more about this when we do action potentials, I think, in the next few lectures after this one. All right? So, there you go. So you remember we said positive ions rush in, at, when the membrane is being depolarized. So when this is happening, how does that sodium ions or the other potassium ions get in and out of the cell? They use channels, okay? And these channels have gates. They have a mechanism that closes and opens the channel, okay? So there are some channels that are gated by voltage. So those are voltage gated channels. It means at a certain voltage, that ion channel will either close or open, okay? So at minus 70, when the cell is at rest, these voltage gated channels are what? Closed. because that's the voltage with which they're closed, and it's resting. All right. So you know, when and then there are other channels that are gated by you know, ligands. That means something must attach to the protein channel to make it contract. I mean, to make it open or shut. Okay? Such as when the impulse comes in, okay, on the neuron itself does not actually touch the muscle fiber. There is actually a small gap between the muscle cell and the, and we will do that in a later lecture about the synapse. So the neuron has to send a messenger to the muscle cell for it to contract. Okay? So that's that ligand. So neurons usually transmit what are called neurotransmitters to pass the message on to the next cell. Okay? So that neurotransmitter at the neuromuscular junction today is going to be acetylcholine. Acetylcholine, ACH for short. Okay? That acetylcholine is attaches to sodium ligand gated channels. Okay? Because when at rest, you've got a bunch of sodium on the outside, a bunch of potassium on the inside. So when that action potential arrives, it will send acetylcholine. It will attach to the ligand gated channels for sodium. Those open up. All right? Those open up, and then sodium gets into the cell. Then it's a positive charge getting into the cell, so that means the cell membrane is beginning to get depolarized. All right? So here's what it looks like. Okay? Okay, so that's what, this is 0, okay? And this is millivolts, and this is minus 70. Okay, normally they go up to plus 30, okay? Plus 30 millivolts. So we are resting, and initial potential comes, and then those ligand-gated sodium channels open up, okay? And then it usually dips below and comes back, okay? So there is a certain voltage that needs to be reached, called threshold, for the muscle cell to contract. If it doesn't get to threshold, then it's not going to contract. Okay, so that's minus 55. Okay, so when the impulse comes in and the ligand-gated channels open up, the sodium-gated ligand-gated channels open up, then it will take the voltage. It must be enough of those channels must open to reach minus 55. Okay. Then the ligand-gated channels will close, and now the voltage-gated channels will pick up, because there's a lot more, you know, voltage-gated sodium channels. Okay, so minus 55 is the voltage at which the voltage-gated channels are sensitive to. Okay, so you see that these, first of all, we stimulate ligand-gated channels. All right. That are attached to acetylcholine, and then we will stimulate the voltage-gated. There's many voltage-gated sodium channels. Okay, so this is the depolarization, and we'll do this depolarization, and sodium is rushing into the cell, and it must reach this threshold first. That's only when the voltage-gated, of which there are many, will open up, and then it will go to plus 30. It always goes to plus 30, it never goes up there. Then at that stage, plus 30, they are potassium channels that are sensitive to plus 30. Those voltage-gated potassium channels will now open up at plus 30, and the ones for sodium will close. Okay? So that, because there's more potassium on the inside, now we are removing that positive charge from the cell, and then potassium now is going out on this side. And this is repolarization. Okay? We're going back to resting membrane potential. Okay? So this is just a short introduction to action potentials that makes you understand these two gated channels, because I really want you to understand a ligand gated channel responds to when a ligand attaches to the ion channel and it opens up. And those are the first ones that open up to reach threshold. Once you reach threshold, we no longer need the ligand gated channels. They close up, all right? And then acetylcholine is recycled, and I'll show you all those events. Then at this stage, voltage-gated sodium channels will open up because there's many of them. There are plenty on the membrane. They open up and it shoots all the way to plus 30. And then that's an action potential. And then usually when repolarization happens, it kind of dips below the resting membrane potential because it's happening very fast in milliseconds. Okay, this is on this side is milliseconds. Okay, and then it comes back up. So that's hyperpolarization. It comes back to polarization. And then the sodium-potassium pump will activate, and that one is stimulated by ATP, it's energy. That sodium-potassium
pump.
So
then the ligand-gated channels only stay open until threshold?
Until threshold, yes. You see what I'm saying? Because the only way the molecule will be sent. The only way neurons can send a message to the muscle is via acetylcholine. So you have to have a ligand-gated channel that will respond to acetylcholine. When that happens, then you no longer need, the signal has already been sent and we've reached threshold. And then an action potential will happen when the voltage-gated sodium channels, of which there are more. Because you only need ligand-gated to go, what? About 20, is it 20, 15 or 20 or something like that, or 15? 20 to 15 voltage difference, so you don't need many. All right? So that's that. And I will talk about both these channels, ligand-gated and voltage-gated. So this is what kind of channel is this? Ligand-gated channel, right? This is the ligand-gated channel. Okay. And these ligand-gated channels are located on the muscle fiber membrane, right? And that part of the muscle fiber membrane is what is called the motor end plate. And we'll talk a little bit more about that when we do the action potential. All right. So acetylcholine attaches, you see how it's closed? Once it attaches, then it opens up and sodium will rush in. So this is a ligand-gated channel. Okay? So that's an easy one. At rest, there's always a bunch of sodium on the outside and a bunch of potassium on the inside. When we depolarize, we just reverse them around, and then sodium is more inside and potassium is more outside. Okay. At the end, we have to use the sodium-potassium pump to change again the concentration to rest membrane potential, where you have more sodium outside and more potassium on the inside. Because the sodium-potassium pump will pump 3 sodium outside and 2 potassium to the inside. All right. So at rest, okay, you have the inside more negative, the outside more positive, right? We haven't stimulated the voltage-gated channels yet. Okay. So the next step is the depolarization that I talked about. Remember when I did that graph for the action potential? Depolarization simply means you've opened the sodium channels. Because sodium is outside of the cell, it's going to rush into the cell. Okay? Just by concentration gradient. You understand? Okay, so that time when sodium rushes into the cell, that's called depolarization. Okay? Depolarization. Alright? And then now the inside is more positive because you've just dumped a whole bunch of positive charges on the inside. Okay? Okay, and then the last step is repolarization, when you go back to resting membrane potential. So in order to remove the positive charges, because the voltage-gated sodium channels have now closed, so that sodium doesn't enter anymore, the only thing you can do is open the potassium channel so that potassium now rushes out to remove that positive charge out of the cell. Okay, so the potassium-gated channels open up, and then the voltage-gated potassium channels open up, and then potassium rushes out, and once again, the inside is
Negative.
more negative, and the outside is more positive. Okay, but do we have resting membrane conditions at this time? Because we've got a whole bunch of sodium on the inside and a whole bunch of potassium on the outside. That's not resting membrane conditions. Okay, so that's when you have the sodium potassium pump. This uses ATP and it pumps out Na plus, 3 of them, and in K plus, 2 of them, okay, to re-establish the conditions at rest. See what I'm saying? So far I've talked about ligand-gated potassium channels, ligand-gated sodium channels, voltage-gated sodium channels, and then voltage-gated potassium channels. Okay, there are other channels, okay, that we will kind of introduce a little bit more when we talk about action potentials called leak channels. Leak channels are not gated at all. They don't have a mechanism to shut and open. They're always kind of open. So the membrane has more leak channels for potassium than it has for sodium. Okay, so potassium kind of leaks out a little bit more. Anyway, just know that there are other channels that are not gated, and those are called leak channels. Okay. All right. So depolarization, repolarization, resting. Okay. Resting, then we have depolarization, sodium in, and repolarization, potassium out, and then we re-establish the rest membrane conditions with the sodium potassium pump. Okay, so that one actually uses ATP energy to pump ions in and out. Okay. All right. So that's that. So this is just simply what's happening with the action potential. It's pretty much what I just talked about at the beginning when I was saying an action potential only occurs when you reach threshold. Okay. So to reach the -55, we use the ligand-gated channels, because the message comes from the neurons via acetylcholine, which is a ligand. All right. Okay. So and then whenever the membrane potential is going more positive, is approaching, it's depolarizing. Okay. So it will depolarize. Okay. And then when it reaches -55, then a whole bunch of sodium voltage-gated channels open up. There's a lot more sodium voltage-gated channels, and then sodium will rush into the cell, and it goes all the way to +30. The inside now becomes positive. Okay. So in the exam, if I was to ask you what is the purpose of the ligand-gated channels, what would you say? What would be the purpose of the ligand-gated channels? The ones that respond to acetylcholine. What is the purpose of that?
To send a message.
Yeah, to send the message. Okay, that's one. What else? So that what?
To reach threshold?
Yeah, so that you reach threshold. So the big thing about them is so that you get to -55, because -55 is the voltage at which those sodium voltage-gated channels will listen to and open up. Then sodium rushes in. Okay, so that's the purpose. And then repolarization, of course, you close up those sodium channels, because at +30 they close up. And then at +30, there are other voltage-gated channels for potassium. Those open up. And then because there's more potassium on the inside, it rushes out. Now it's removing that positive charge, so that inside it's more negative once more. Okay? So that's the repolarization. Repolarization is that we're going back to polarization. Depolarization means we are undoing the polarization. Remember, we said polarization is simply separation of charges. That's it. You've got more positive on one side and more negative on the other side. And that's all. So when we depolarize, we undo that. When we repolarize, we re-establish rest conditions. That's all that is. Okay. So action potentials will occur in an all or none fashion. That means you have to reach threshold first. When those ligand-gated channels open, when acetylcholine attaches, you have to get to minus 55. If it gets to minus 60, what happens? Nothing. Okay? It doesn't, a little bit of action potential. Like I always say every year, it's like being pregnant, right? Either you're pregnant or you're not. You can't be just a little bit, pregnant, just a little bit, So it's either or. That's the same thing with this. Either it happens or it doesn't. If it doesn't reach minus 55, nothing happens. It has to reach minus 55 or go above minus 55, then it happens. Okay? So it's all or none. So the stimulus will produce an action potential only if it reaches or goes above minus 55. That's it. And then, like I said at the beginning, the way the action potential travels along this nerve fiber or along the muscle membrane, fiber membrane, is via propagation. It means this part is an action potential, then the next part, then the next part, then the next part, and that signal travels along. And that is propagation. Okay? So action potentials, all or none, either happens or doesn't. Okay, so this is just an action potential.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium-ion channels are closed, and the inactivation gates are open. Voltage-gated potassium-ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium-ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
So that little dip that goes under is weathered toward the end of the graph. That's called hyperpolarization because you've separated the charges a lot more than minus 70. That comes back though when the sodium-potassium pump reestablishes the resting conditions, right? Okay, so very good. So the understanding how the action potential is sent along the nerve fibers is important because that's the only reason you will stimulate the muscle to contract. So since we're talking about muscles, it's the somatic neurons that are going to stimulate them. Somatic means muscle somites because somites are the structures in the features that will become muscle. So then anything to do with the muscle is somatic. Okay, so the somatic nervous system, motor, because you're causing the muscle to do something, so it's a motor. Okay, whenever something comes from the nervous system and it's sent to effectors like muscles, glands, those are motor neurons because they're causing those things, those effectors to do something. Okay, and when you pass impulses from the periphery to the nervous system, that's sensory because you're sensing what's happening around. Okay, so this is motor because it's causing the skeletal muscles to contract, to do something. All right. So again, the axons are the nerve fibers because a nerve cell has got a cell body which has got dendrites like so. Okay, and then a long axon. Okay, like that. So the axon is where the action potential is propagated like that. And then it's traveling down the nerve fiber. And then nerve fibers they branch profusely, a lot. You can have thousands of branches on one nerve fiber. Now, why is that important? Because every muscle cell, has a neuron, branch that will control it. So you can imagine your muscles, your thigh muscles. How many muscle cells do you have in your thigh muscles? Like a ton of them, a lot. So you can't have one neuron coming to, you know what I'm saying? Each of those cells, it's just going to be too much wiring. It's impossible. You'll be all neuron. The whole body will be neuron just to stimulate thigh muscles alone, let alone the calves and all that. So the one will branch to many, to thousands, of branches, and then each one will go to a cell, a muscle cell, a muscle cell, a muscle cell. And that's what is called the neuromuscular junction, the neuron and the muscular junction. And like I said, they don't touch. That's why you need that messenger, the acetylcholine that is sent to stimulate the ligand-gated channels, right? Because they don't touch. Well, what's the significance of not touching? Why don't they just touch? It's so much easier, right, when they are touching.
Because the muscle would constantly contract.
Yeah, the muscle, once it's physically adjoined to the neuron, if you have a message, a false message, something excites the neuron, each time the muscle will contract, even when it doesn't need to. So if they don't touch and you send a messenger, you have more ways of controlling that. If a nervous system is stimulated, something comes out, you could kill the acetylcholine, you could break it down before it gets here so that nothing happens. You see, you have more ways of controlling it. So that's why it doesn't touch, okay? Like I say, there's always a reason for these things, in the body. Nothing ever just happens because it's kind of cool. It happens for a good reason, okay? So that's what it is. So all this is because of that, and because muscles have many cells. So you need many branches for that. So now we're going to talk at the events. What is the physiology happening at the junction of the neuron and the muscle cell? Right? Mario? Okay, so the neuromuscular junction is formed at the junction of the neuron and so those neuron branches are neuron terminals, because that's where it terminates. And each terminal will terminate on a cell. They have vesicles in them. When it branches, like so, it's got a little knob right there at the branch. Okay? So that, and this is a muscle fiber. Okay, muscle fiber. They don't touch it. Okay, so in the synaptic knob, which is the presynaptic membrane, because this is pre-synapse. Synapse is that functional connection between the two, right? It's full of vesicles that have acetylcholine in them. Okay, so when the impulse comes along here, it's going to stimulate exocytosis of those vesicles carrying acetylcholine. Okay, so that's what we're going to talk about in a little bit on the things that happen or the events at the neuromuscular junction. Okay, so all what the impulse is going to do when it reaches the end, it's going to open calcium channels, and then the calcium, when it goes into the synaptic knob, it's going to attach to those vesicles and cause them to attach to the membrane for exocytosis to occur. So that's what. So the impulse will come in, open up those potassium, those are voltage-gated calcium channels. Sorry, calcium. Ca2+. And this is the calcium that attaches to those vesicles. You see these synaptic vesicles carrying ACH, acetylcholine, the ligand that we're going to need to attach to the muscle. And then when exocytosis occurs, so this is the junction. Acetylcholine is going to be sprinkled into that gap, into the synaptic cleft. And then it will go and attach where? To those receptors right here. Those sodium ligand-gated channels. Okay? So the part of the muscle fiber or muscle cell membrane that has these sodium ligand-gated channels That's what's called the motor end plate. Okay, do you see that? I'm saying a lot of things here, but do you follow that? No. This is the synaptic knob, and inside of it is got synaptic vesicles with acetylcholine, right? You see what I'm saying? Like that, and that's the branch. This is the branch. Okay, I just blew it up. I just blew this up. You know what I'm saying? I blew this part up to here. Okay. So the impulse gets to the end of the axon, and it will open calcium channels, and we'll talk about that in a little bit, step by step. Okay, calcium channels will cause the synaptic vesicles to attach to the synaptic knob membrane here, and then release the acetylcholine into the cleft. Okay, and then the acetylcholine will attach to the ligand-gated sodium channels. Okay, so all we're saying is that the part of the membrane on the muscle that is these ligand-gated sodium channels is called the motor end plate. That's all. Okay, so know what the motor end plate is. Okay, and know what the neuromuscular junction is. All right. Okay. And know that these axons, they branch profusely into many branches. Okay? Because we simply couldn't have one neuron controlling one muscle cell. There'll just be way too many neurons. It just won't be enough. Okay? So, gotta know these terms now, all right? So we can go into, you know, like I said, the neuron doesn't, the neuron terminal doesn't touch the muscle cell. It comes very close to it, but it doesn't touch, okay? And we said the reason why is so that we can control the stimulation of muscle more, okay? And the gap, the physical gap between the axon terminal and the muscle cell is called the cleft, synaptic cleft, okay? And the connection between the two, the neuron and the muscle cell, is functional, is not physical, okay? That's what's called a synapses, okay? Okay, and the gap is the physical gap between the two is the cleft, synaptic cleft, okay? So this is a picture showing, see, so this part from here to there is the motor end plate, because that's what has those ligand-gated sodium channels. And this is the synaptic vesicles carrying what in them? What are the synaptic vesicles carrying? ACH, acetylcholine. And then exocytosis, remember when we did cells and we did movement and transport, we did exocytosis, so we know what that is already. Okay? So, and this is a picture of a neuromuscular junction. Alright? So the calcium comes out, and then we'll attach, we'll cause these vesicles to attach here, and then sprinkle the acetylcholine in here, into the cleft.
But doesn't calcium go into the neuron?
It goes into the, into here. Yeah. So on here will be, calcium channels will be here. All these are calcium channels, okay? So all this interstitial fluid has got calcium in it, you know, sodium, all these ions. So whichever gate opens will let whatever kind of ion opens. So calcium will need to get in here. Okay, so calcium plus, calcium 2 plus, all here will cause these vesicles, the synaptic vesicles carrying ACH, to stick to the membrane here, to the presynaptic membrane, to cause exocytosis. So it's the calcium that causes the exocytosis when those vesicles release the acetylcholine into the cleft. Remember, the synaptic cleft is the physical gap between the two. So this is the cleft, and then acetylcholine is sprinkled in here, and then on the motor end plate, we have those ligand-gated sodium channels waiting, and then they will attach to the acetylcholine and open up, so that we reach threshold. We go to minus 55, boom, we have an action potential. If not, nothing happens. All right? So the trick is to have enough sodium-gated, ligand-gated sodium channels to open to reach minus 55. Because once you do that, then a whole bunch of voltage-gated sodium channels open up. Okay? again another picture. You can see some axons, and then you can see the neuromuscular junction here with these synaptic knobs and whatnot. Okay, and you can see each one goes to each cell, each muscle fiber. All right. So this is what's happening at the neuromuscular junction here. Okay. So we have the first step. So we're going to go through each of these steps. There's like 9 steps that happen here. And I want you to know the steps. These are the steps that you guys were in the module. We're supposed to lump into the leg phase. Because what happens is that, let me just draw this for you a little bit. Okay? The muscle is at rest, right? Okay. And then, boom, 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 it goes into the contraction phase. Contract. And then, boom, 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 it goes into the relaxation. It relaxes, right? So what we are talking about is that before the impulse comes, let's say we apply the impulse right here. Boom, boom. The impulse comes in. There's a time between arriving of the impulse and actually when the muscle contracts. Okay? The muscle doesn't just go boom, contract immediately as soon as you stimulate it, because it's got to get ready, right? It's just like when you're doing a 100-meter race, they say set. They don't just blow the gun and, you know what I'm saying? Because that will cause a bunch of false starts. Don't you think? Yeah, so you say set. So what's happening in the leg phase is the set part, where you get the impulse coming in, the calcium is opening up. So those are the things we're supposed to lump into the leg phase. The calcium comes in, the vesicles attach, then when the, the acetylcholine is sprinkled in here, we're still in the leg phase, right? Because the impulse hasn't even gotten here yet. And then when they start to depolarize, that's when we start to have the contraction, because then it reaches minus 55 and then we have the contraction. So all those events that happen prior to the voltage-gated channels open is the leg phase events. Okay? So the first step is that the impulse comes in. So this is part of the leg phase. The impulse comes in, leg, L-A-G. Then it opens up this calcium, voltage-gated calcium channels. Okay? And then calcium goes into the synaptic terminal, or the synaptic node, which is here. Okay? So that's number 1. I hate this, the way they set it up, so that, okay, so number 2, what happens? The calcium will attach here to this synaptic vesicles carrying with them acetylcholine, and then it will cause exocytosis. You all know about exocytosis, right? When, the vesicle attaches to the membrane, and then the contents of the vesicle are externalized. That's it. So you guys know all too well about that. Okay, so it will cause exocytosis. All right. It will initiate the release of the neurotransmitter. All right. Okay, so number 3. There you go. So then, after they become sticky and they stick to the membrane, the exocytosis occurs, and acetylcholine, you see, acetylcholine is acetyl and choline together, so they are released, all right, into the synaptic cleft, and they will, because they are so close together like that, go and attach. So this is the motor end plate. You see the part with the sodium ligand-gated channels. And then once acetylcholine attaches to the sodium ligand-gated channel, what happens? They open up and sodium starts to rush into the cell. Okay, so that's number 3. Sodium starts rushing into the cell. Okay. Anyway, so number 4. What's happening in number 4? The acetylcholine that has been released is now attaching to the ligand-gated channel, and sodium is now going in. So we are beginning to depolarize now. We're going towards minus 55. Okay? Because the purpose of the ligand-gated channels is to go to minus 55, which is threshold. Okay? Once we get to minus 55, then the voltage-gated channels take over, because they're sensitive to minus 55. it will open up a whole bunch of voltage-gated sodium channels. Then we actually close, and I'm going to show you later on, we will close up those ligand-gated channels. We will break down the acetylcholine and recycle it. Okay? Okay, so that's that. So acetylcholine diffuses in. Okay, so number 5, what's happening at number 5? We're getting the sodium coming in, and then we're reaching threshold. Right? Remember, we have to reach minus 55 before those voltage-gated channels open. So ligand-gated channels open up, and sodium gets into the cell. All right? So calcium channels open up, number 1. Calcium channels open up, calcium rushes in, and then it attaches into these ligand, to these vesicles. Okay, just a quick recap. Once that happens, number 3, the vesicles become sticky and exocytosis occurs, and then acetylcholine comes out. Number 4, the acetylcholine will diffuse through the gap, the synaptic cleft, and attach to these ligand-gated sodium channels. Okay, once that happens, they open up, and then number 5, it's rushing in. Sodium rushes in, right? And then we're now approaching threshold, because now we're beginning to depolarize. All right? We have to reach threshold if we're going to have an exit potential anyway, right? So enough of those ligand-gated sodium channels open up so that the membrane potential goes to minus 55. All right? So once that happens, we've reached our threshold, right? We don't need them anymore. We don't need the ligand-gated channels anymore because there's only a few of them anyway, okay, on the motor end plates. So number 6, we shut them down, and we will, and then the acetylcholine detaches from the receptor, and then it closes. So acetylcholine is removed from the receptor, the ligand-gated sodium channels, and it closes. Because at this point, the voltage-gated sodium channels are taking over, right? Because they opened up sensitive to minus 55. Okay, so that's that. So we close up those, we remove acetylcholine. Okay. Once acetylcholine is removed from the, ligand, we're going to reuse it. Because imagine if we're doing this all the time and we're just wasting away the acetylcholine, we'll soon run out and we won't have the resources to make new acetylcholine. So we have to recycle it. So these events now are aimed at doing that. Because we've already stimulated the muscle, it's doing its thing now. Okay? So we have it also on the motor end plate. Remember I said we got the sodium ligand-gated channels. Okay. We also have another enzyme on the motor end plate called acetylcholinesterase. Guess what acetylcholinesterase does? Yeah, breaks down acetylcholine. Okay. So we have another. So the motor end plate has got those 2 channels, proteins on it. One is a ligand-gated sodium channel. The other is acetylcholinesterase, an enzyme that breaks down the acetylcholine into choline and acetic acid, because that's what makes acetylcholine. Acetic acid contributes to the acidity that happens on your muscles and also accumulation of lactic acid via anaerobic respiration. It causes that acidity, that burn. You know, when you're working out, that burn you get at the gym is that nice burn. It's because of this acid that comes out. You know what I'm saying? When you're working out. So the acetic acid goes back, it's remetabolized, it's used up again, and then it goes into the blood. It will come back again here. So the number 7 step is pretty much acetylcholinesterase breaking down acetylcholine into choline and acetic acid. Acetic acid just diffuses into blood, and then it's recycled again. And then the next step is to do with the choline, right? Boom. So the choline now remember when we did transport and we did symport, uniport, and antiport. What does a symport do? It will transport two molecules in one direction, right? So what happens is that there are sodium channels, okay, right there at the presynaptic membrane on the synaptic knob, okay, that when they're transporting sodium into the synaptic knob, they will piggyback choline with them. So choline is sympported with sodium channels. You see what I'm saying? So the sodium channels, and they will piggyback, that transport will piggyback acetylcholine, move at the same time. So acetylcholine moves back into the synaptic knob. And this goes into the blood, acetic acid, and then it will come back again in the blood, and then we're now going to reassemble that acetylcholine again. Okay? You see, so the events at the neuromuscular junction are nothing more than the impulse coming in, calcium gates opening up, voltage-gated calcium, causing that exocytosis with the vesicles, synaptic vesicles, and then it sprinkles into the knob, attaches to the ligand-gated channels, sodium ligand-gated channels, open up, reach threshold, and then at that time, the voltage-gated sodium channels take over, and then we close the ligand-gated channels. Then we have to recycle the acetylcholine. That's all they're doing. Okay, so think about just that. Think about getting the exocytosis to occur with the calcium. Think about using the acetylcholine to open the ligand gates for sodium to reach threshold. And once you do that, you no longer need the acetylcholine. We have to break it down and recycle. That's all you're doing. So if I ask, usually I ask all the time, events at the neuromuscular junction. And it's like a 5-point question with maybe 3 or so bonus points. Okay? So you should Google that up and you just think about what you're doing, what you need to do. So then the acetylcholine is imported into the cell, into the synaptic knob with the sodium channels. Symport. Okay? They go in the same.
So when it goes back with the sodium, it's only the choline that goes in?
Only the choline that goes in. The acetic acid just goes into blood, and then because it goes into circulation, it's gonna come back into the synaptic knob eventually, because the blood will be full of it, and then because you're gonna need that acetic acid again to because the acetic acid is easy to transport in the blood. It just goes into the blood and comes back again to the knob, and the knob through its in fact, this kind of goes to the liver and is then made back into glucose again, because it carries 3 carbon atoms, and then you add it together, you have 6 to make the glucose. But because in here there's metabolism going on, and you have acetic acid anyway, you can create it, so you don't need to import it to waste energy trying to shove it in when you can create it in the cell already. So you create that, so you don't need it. But it's the choline that's more expensive, so we gotta bring it back. You know what I'm saying? So we bring it back in with the symport, the sodium symport channel, to move it in. Okay. So when you think about this process, don't think about 9 steps. Think about what you need to do. You just need to get those vesicles to exocytosis. And the only thing that causes is the calcium. So you need to open calcium channels. So that's what the impulse does. And then when you're done with that, it diffuses through, attaches to the sodium ligand-gated channels. And then once you get to threshold, you don't need the acetylcholine anymore. You don't need the ligand-gated channels anymore. So you have to break down the acetylcholine and recycle. Okay? And if the muscle cell is not doing its thing, contracting and so forth, or whatever, and then you are processing again more acetylcholine, then you symport the choline in. The acetic acid is already made in the synaptic knob, so you don't need to transport it back in there. You just let it go into blood, and it's taken care of by metabolism. And then you just assemble the acetylcholine, and you wait again for the next impulse, right? And so in the cell, in number 9, what are we doing? We're using the choline that we just imported in, plus the acetic acid that is we form in the metabolism of the cells in here anyway. And then we form acetylcholine again. We pack it into the vesicles. Okay, so the Golgi body and all those are involved, right? Remember those things, the Golgi packages and, assembles things and stuff like that. And then it will be packaged by Golgi body, and then the vesicles wait for the next impulse. Okay, that's it. Those are the 9 steps. Okay. So we recap again. Impulse comes in. What happens? Calcium channels open. Alright? And then what is the purpose of the calcium? Attaches to the vesicles, causes the vesicle to stick to the membrane and form, and then exocytosis happens. Then when that happens, this acetylcholine is sprinkled into the cleft. Then it goes and attaches onto the ligand-gated sodium channels. Right? They open up, and then sodium starts getting, and then the membrane is starting to depolarize. We reach threshold, voila! We no longer need those ligand-gated channels, because at that point, a whole bunch of voltage-gated sodium channels open up. So at that point, then they close, all right, and then we remove the acetylcholine from the receptor, from the ligand-gated channel. Then there's also another enzyme waiting on the motor end plate, acetylcholine cholinesterase, and we break it down. We take the choline back in, we recycle it back in, all right, via a sodium symport. And then the acetic acid just goes into blood, and then it's metabolized by the liver and so forth to make other things. We already make acetic acid in the cells at the node, so we don't need to transport it back in, right? And then we use that acetic acid plus the choline that we imported in to form acetylcholine. Acetylcholine waits in the vesicles, and the Golgi body will package it up in weights until another impulse comes in. So all those are events at the neuromuscular junction, all right? And most of those events fall into the lag phase, okay? Because really the real action happens when you reach threshold. That's when you start to contract. So all those events, most of those events here are grouped into the lag phase, okay? And again, we always show this picture here that shows how these systems interact, and you should always look at it to see how your system interacts with the rest of the other ones. And that's it, guys. So today we finished nice and early. So we're going to, I'm going to go set up, and if you want, we can start early and finish early. We can start at 2:15, or you want to start at 10? You want to start at 10 so you can start?
10.
Yeah, you start at 10, guys. I knew you'd start at 10. Okay, guys, thank you so much. Go and study, and then we'll see you at 10, okay?